I'm very happy to introduce Ross Carlson, who is a professor of chemical and biological engineering at the Center of Biofilm Engineering and Thermal Biology Institute at Montana State University in Bozeman, USA. So he has a degree in biochemistry, microbial engineering, and chemical engineering from the University of Minnesota. His research interests span fundamental biology, including the design rules of life, environmental microbiology, like nutrient cycling in geothermal hot springs, and medical microbiology, including chronic wound biofilm. So I will let you present your topic.
Great. Thank you very much. Thank you, Sabine. Thank you for the organizing committee for the opportunity to speak. I do appreciate it. So I'll be discussing topics that are relevant to the previous two talks. So I'm also interested in microbial physiology. I'm also interested in biophysical constraints. So today's talk is going to be looking at the cell geometry. So we'll be looking at the actual physical limits of a cell. And what do the physical limits of a cell, how do they constrain phenotype? So most interactions, if not all interactions that cells have with the environment occur via the surface area of the cell. So things like acquiring nutrients, communicating with other cells via quorum sensing. These all occur across the surface area of the cell. The surface area sets that physical limit of the existence of the cell, and we feel that places major constraints on its phenotype. So what I'll be discussing today builds on some concepts that were published in the past, but I think it's an area of research that is really underexplored, and hopefully after the talk today, I'll get some more people excited in the field. So the test case for this particular study is two common strains of E. coli. So we're looking at K12 strains, the blue strain, so the theme throughout the talk, the blue strain will be MG1655, and the red strain is going to be the NCM3722. So common K12 strains, these have very similar genomes, very similar genomic potential, but these cells have different properties. So if you look at the upper left here, what we're looking at are 30% differences in the specific growth rates of these cells. These are the mu max values, and we see about an 80% difference in the growth rate at which overflow metabolism occurs. I'm guessing that most of us are familiar with these concepts, but I'll just review them quickly. So a complete oxidation of your substrates, we're looking at things like glucose and oxygen here. So complete oxidation, the cell would oxidize the glucose all the way to CO2 and water. When we have an overflow metabolism, what we're looking at is a partial oxidation. So overflow metabolism is sometimes called the Crabtree effect or the Warburg effect, depending on which organism you're looking at. But what you're looking at is the consumption of glucose and oxygen, so an electron donor and electron acceptor. You get carbon dioxide and water, but you also get some partially oxidized byproducts. So major phenotypic shift. It's an area of a lot of scientific research. And these two very similar strains of E. coli engage in this overflow metabolism at very different growth rates. In fact, this NCM strain switches to an overflow metabolism at a growth rate that is greater than the mu max of MG1655 when these are grown on salts media. So the hypothesis is why, or the question is, why are these parameters so much different? The hypothesis that we'll explore today is that it is related to the physical dimensions of the cells. So the diagram here in the lower left is looking at the surface area to volume ratio of these two strain types as a function of growth rate. This includes both defined media and rich media, so we'll see that the growth rates in this case actually go up to higher rates, higher than what I'm listing here. The reason for that, again, is this is salts-based media, this is salts and complex media. So we see substantial, we see significant, I would argue, differences in the surface area to volume ratio of these two cell types as a function of growth rate. I've got some illustrations here of MG1655. These are set up in a quantitative manner so that we can appreciate how the dimensions of these cells change as a function of growth rate. So the length of the cell and the diameter of the cell change as a function of growth rate. If we look at the NCM strain under these same growth rates, we can look at their dimensions. And again, these are quantitatively accurate. And we can see that there is a substantial difference in the dimensions, leading to a substantial difference in the surface area to volume ratio. So hypothesis here being the surface area available for acquiring nutrients, the surface area available for important processes like respiration are influencing the phenotypes of these cells. So we're interested in two major parameters. So one is the surface area to volume ratio. The second parameter that we're interested in is molecular crowding. So molecular crowding refers to the physical space that molecules occupy. In this talk, we're talking about the membrane. So this is a cartoon looking at the membrane of E. coli, a typical gram-negative cell. We have an inner membrane and an outer membrane. For the balances that I'll be discussing today, we're looking at the constraints of the inner membrane on the cell's phenotype. So the inner membrane hosts a wide variety of proteins. These proteins are responsible for things like substrate transport. So for the cell to acquire resources like glucose, it needs to express transporters in the inner membrane. And then to run respiration, the cell needs to express enzymes associated with the electron transport chain. So we're looking at a typical arrangement here. And if the cell wants to use that proton motive force to generate ATP, it needs to express ATP synthases. So what we're seeing is a bunch of proteins here. So there's a limited or a finite amount of surface area, and there is a finite capacity of this membrane to host proteins. So you can't load this with 100% protein. The current estimates for the area fraction of the inner membrane that can host proteins is somewhere between 20 and 30%. So this represents a fairly substantial knowledge gap. And I think it's related likely to some of the complex properties of enzymes that exist in these membranes. So they tend to have both hydrophilic and hydrophobic zones, which makes the analysis a little more complicated. So what we'll be looking at today is how do the intersection of surface area to volume and the membrane capacity to host proteins, how does that theoretically influence the phenotypic limits of a cell? So looking back at these notions, so this notion of surface area to volume constraining phenotype, if you look at the literature, there's actually quite a few people saying that surface area is not a constraint on phenotype. And the basis of their argument is this plot that I have on the left. So surface area to volume decreases with increasing growth rates. People interpreted this plot as if the surface area decreases with growth rate, certainly it can't be limiting the capacity of the cell. There must be some other major constraints. But looking at just the surface area to volume misses a big part of the story. If you actually look at the amount of protein that is hosted in the membrane as a function of growth rate, the amount of protein hosted in membrane actually goes up with growth rate. So surface area to volume decreases, but the capacity of the membrane to host protein goes up. And we're hypothesizing that has to do with the curvature of the membrane as the cell gets larger. We're hypothesizing that the membrane has a larger capacity to host proteins without losing its structural integrity. So surface area to volume decreases with growth rate, but the cell actually increases its catalytic capacity of the membrane by hosting more proteins in the membrane. So we're interested in phenotypic limits on this. So we've adopted approaches much like the previous talk. So we're looking at constraint-based modeling. We're looking at stoichiometric modeling. In this case, we'll be using flux balance analysis. So how do we integrate this concept of a limited surface area to volume and a limited macromolecular crowding into an FBA type format? So what we did is start off with each of the enzymes that's associated with the membrane. What we can do is we can calculate the area required for each of those enzymes as a function of things like the flux. So in this case, V is going to be your specific flux. The gamma in this case is the area, surface area of the membrane required per enzyme. We have the kcat value here, so the turnover rate of the enzyme, and then we have a saturation factor. The rest of the terms here then are just conversion factors. And I want to highlight that what we're looking at is a surface area constraint. It's not a volume constraint. So this is a bit of a difference from a number of studies that are currently in the literature. So we're looking at how much area each enzyme occupies in this inner membrane. This is in contrast to the entire volume of the enzyme. Some folks have been looking at the total volume as a proxy for the total investment. If you look at the relationship between the enzyme volume and the surface area requirement, there isn't a strong relationship there. We're arguing that this membrane surface area is a unique biophysical feature of these enzymes and should be analyzed as its own parameter. So again, we're looking at surface area per enzyme. What we can then do is sum that up over all of the active enzymes. So sum up over all of the enzymes that are active in the membrane. We are doing some unit conversions here. So we're going from a specific growth rate to a volume, and then from a volume to a surface area. So there's a number of conversions such that this equation is the fraction of the membrane surface area that's being occupied by active enzymes. For this conversion, we need a surface area to volume conversion. And of course, we know that changes with growth rate. So we've integrated in a growth rate dependent surface area to volume ratio such that we can account for those changes in cell geometry as the cell grows at a different rate. We can collect terms here. And what we end up with is a linear constraint. So what we have is a resource budget. And this resource budget is the capacity of the membrane to host proteins and accounts for the surface area that's available. That resource is consumed by active enzymes, and that resource is consumed by faster growing cells. So as cells grow faster, there's less surface area per volume, less surface area per gram of cell dry weight. So that's a bit fast. We do have a preprint out. If you're interested in the details and all of the units, you can find that in the preprint. I'll show that later on in the talk as well. So we're implementing this constraint into our FBA models. What does it look like? So we can calculate budgets. These budgets are a function of the assumed macromolecular crowding. So for instance, a typical magnitude would be, with the units we're using, is about 13. So with the appropriate conversions, what we're looking at is an area per gram of cell dry weight that is available for active enzymes. What we can do is integrate then a cost into each of the reactions in our metabolic model that are associated with the membrane. So this happens to be the glucose transport reaction. And this is a term that's scaled for the parameters of a cell. So for each flux through the system, we're consuming some of that available surface area, and that's being taken from that constrained budget. So next thing we'll look at briefly here is what is growth. So this is informing how we are using this constraint to analyze the phenotype of the cells. So growth is a combination of anabolic and catabolic processes. Anabolic processes are things like the production of macromolecules. So this is the production of the physical components of biomass. Typically about half of your glucose going into an E. coli cell is going to go into macromolecules. The other half of the glucose that goes into the cell is going into a process known as maintenance energy. So this is an implicit process. It's largely a black box process, but this is a large collection of cellular processes that consume cellular energy, and we could think of those processes as a fight against the entropy of the universe. So these are interpreted as processes that maintain ion gradients, processes that maintain proteins, and things like that. So taking this notion of splitting our metabolism into anabolic and catabolic processes and applying this constraint associated with a limited surface area and a finite capacity of the membrane to host proteins, we can define a solution space. And we happen to plot this in a format that relates to a lot of common questions. So what we're looking at is the relationship between rate and yield in E. coli when it's constrained by a specific membrane surface area. So this is our constraint that looks at the limited surface area available to the cells. This was plotted by using FBA analysis and by setting a matrix of catabolic and anabolic processes. So what we did is we set growth rates, so set fluxes to macromolecule synthesis, and we set fluxes to maintenance energy. And then for each of those simulations, what we did is we minimized the consumption of glucose to achieve those two processes for growth. So we're looking at the constraint again of the membrane surface area. You'll notice there are some ridges in this surface area or in this solution space. These ridges correspond with transitions in the efficiency of the electron transport chain. So as we go through different levels of limited surface area, the most efficient use of the membrane changes, the most efficient use of the available electron transport chain changes. Another thing of interest to note here is if we're at a high yield, we are at modest to low growth rates. So at high yields, the limited surface area is being used for high efficiency electron transport chain processes. So the cell is making a lot of ATP from a limited amount of electrons. On the other extreme here, we have cells that are growing very fast. In this case, these cells are using the limited surface area for lots of glucose transporters. The yield is low, such that we don't have efficient electron transport chain components there. And you can see that there's a continuous distribution between those two extremes. Things to note, people often like to argue that E. coli is optimizing either rate or yield. If you look at E. coli, MG1655, typical mu max on Salt's media is about 0.7. Typical yield is about 0.4. So, based on this analysis, E. coli isn't maximizing rate or yield. It's maximizing, or it's existing somewhere in the middle. It's existing in the middle because of this demand for maintenance energy. So we wanted to apply this theory to experimental data, and we've done a number of different analyses. The hypothesis for this plot is that experimentally measured fluxes reflect this membrane surface area constraint. And we've got 8 sets of data here, 6 of these from my lab, 2 of these from Terry Wa's group. And what we're doing is we're taking experimental data and compressing them into 3 vectors. So we're looking at the growth rate, we're looking at the glucose consumption rate, and we're looking at the acetate overflow rate. The hypothesis being that the magnitude of those 3 vectors represent or reflect this membrane constraint on the system. So for those 8 different data sets, 2 of them with the NCM strain, 6 of those with the MG1655 strain. If we use a Euclidean distance to map the relationship between those 3 vectors to these solution spaces confined by this membrane surface area constraint, what we see is that there is a certain window, and this window is from 5 to 7% of the available surface area being utilized by the enzymes. So our interpretation of this is the experimental data does represent a constraint, and that constraint maps to a relatively narrow range of potential surface areas. And again, in this case, it's 5 to 7% of the membrane being utilized. Keep that number in mind. So we're going to use a couple of different analyses with experimental data, and we'll see very similar numbers using these different analyses. So for the other analyses, what we did is a literature review. And with these literature reviews, we gathered a consensus data set for the growth of MG1655 and the NCM strain. Top plot here, what we're looking at is the glucose uptake rate as a function of growth rate. So these are across 5 different studies. And then the bottom plot, what we're looking at is the acetate overflow rate as a function of growth rate. So we did linear fits of this. We call this our consensus data. And what we want to do is interpret this consensus data using this membrane surface area constraint. So I'm going to walk you through an interpretation of this consensus data. What we did is we took the consensus data, we used that as constraints for our FBA analysis. So we set our growth rates based on the consensus data. We set our glucose uptake rates based on the consensus data. We set our acetate overflow as a function of the consensus data. We applied this constraint based on a limited surface area, and then we asked the model to maximize the capacity of the model to produce ATP, so maximize the production of maintenance energy. What we're looking at is a typical profile here. So we've got our maintenance energy capacity here as a function of growth rate while being constrained for those consensus fluxes. You'll notice each of these, this is predictions, each of these have error bars. And the reason for that is for each point, we did 100 simulations. For each point, we did 100 simulations where we set the consensus fluxes. We perturb the consensus fluxes within their confidence range and then resolve the problem. So we're looking at not only the result of the consensus fluxes, but also a sensitivity analysis. So I'll walk you through our interpretation of what these plots represent. So at low growth rates, the consensus fluxes represent a scenario where these cells can both increase the maintenance energy generation and the growth rate. So you have a positive flux here. So the cell can increase both of those fluxes without penalty. We get to the middle region here, and it's more or less a zero slope. In this case, the cell can increase its growth rate with no penalty in the ability to generate maintenance energy. And then in the last area here, we have a negative slope. So our interpretation of the negative slope means the cell has the capacity to increase its growth rate, but the only way it can increase its growth rate is through a decrease in maintenance energy generation. So what's happening here is the cell is running out of surface area available for maintenance energy flux generation. And in order to grow faster, it would actually have to decrease its capacity for maintenance energy generation. So we're looking at a Pareto front that's defined by the limited surface area. So what we can then do is look at that for a bunch of assumptions around the protein crowding. So we're looking at from 4% to 8% of the membrane being occupied by proteins. For each of those, we ran a different set of simulations, and just to highlight what's going on here. So the simulations in the red box are all using the same experimental consensus fluxes. So all 5 of these data sets represent the same growth rate, same glucose uptake rate, same acetate overflow rates. The difference is we are applying different membrane crowding constraints on it. If there's less area available, the cells can't make as much ATP. If you have more area available, the cells can make more ATP. So we're seeing about an 8-fold difference in our predicted capacity for maintenance energy based on the assumed crowding. Now what I want to highlight is the experimentally measured mu max for our MG1655 strain. So its mu max is about 0.7. 0.7 corresponds with the molecular crowding assumption of 7% of the membrane being occupied by protein, reaching its Pareto front. So our interpretation of mu max is the cells will increase their growth rate until they run out of surface area that can support both an increase in growth rate and an increase in maintenance energy generation. So again, Pareto front defining what our mu max is. You can do the same thing with the NCM strain, and we see a very similar trend. So again, we've got consensus data for this NCM strain. It grows much quicker than MG1655. For that consensus data, we ran our simulations, assuming between 4 and 8% of the membrane is available for enzymes. And then what we looked at is the Pareto trade-off. So at what growth rate do we see a trade-off in the ability to generate macromolecules and the ability to generate maintenance energy? And we see that trade-off occurs somewhere between area usage of 7 and 8%, and that corresponds again then with the mu max that is experimentally measured. So our interpretation is mu max is limited by surface area. Mu max occurs when the cell is forced to make a trade-off, a Pareto trade-off between growth rate and maintenance energy. So that's mu max. Now, what about the respiration efficiency of these cells and the PO number? So if you're familiar with this concept of PO number, it's a way to quantify the efficiency of the electron transport chain. E. coli has a flexible electron transport chain. It has a number of different components that can be mixed and matched in different mixtures that result in PO efficiencies ranging from 0 to 2. PO number, of course, being the number of ATPs that can be made per half an oxygen, so per single oxygen molecule. The theoretical range, again, is 0 to 2 in E. coli, but what you'll notice experimentally is they don't operate at a maximum efficiency. They operate at a value of about 1.1. Question is, does this membrane usage provide us a theory for interpreting that number? So what I'm looking at here is experimental data and theory. So the colored data points here are from experimental data sets. So we're looking at 3 different proteomic studies. We took that proteomics data and converted it into an experimental PO number. We're seeing that experimental PO number is ranging approximately from 1 to 1.2. The blue points here represent our predictions using this consensus data and using the constraint based on the specific membrane surface area. What we see is our predictions are consistent with experimental data. So the cell is not operating at the most efficient use of the electron transport chain. It's using a value about half of the maximum. It uses that value because it runs out of surface area. So if it wants to balance maintenance energy generation with growth, it can't use the highest efficiency system. It has to use something in between. If we look at a fit of the predicted PO number as a function of growth rate, there's actually an inflection point. And that inflection point corresponds with the initiation of overflow in our predictions and the initiation of overflow metabolism experimentally. So by looking at the PO number and the inflection of the PO number as a function of growth rate, we can quantitatively predict when overflow metabolism starts. This prediction is, we say, about 0.5 inverse hours. If we look at the experimental data, it's starting to occur somewhere between 0.4 and 0.5. So again, consistent with the data. We also see that experimental biomass yield as a function of growth rate maximizes at about that transition point. So our interpretation is that the cell becomes limited for surface area. When it becomes limited for surface area, it transitions to this overflow metabolism that allows it to continue to increase its maintenance energy flux while increasing growth rate until it gets to the mu max point. Do the same analysis with this other strain. Again, this other strain having different growth rates, different overflow properties, and different geometries. We account for those different geometries integrated into our analysis. We have our predicted PO number as a function of growth rate, and we have our experimental PO number as a function of growth rate. This is based on proteomics data from Mori et al. We notice, again, an inflection point here, this inflection point quantitatively predicting the onset of overflow metabolism. So our interpretation here is this is a critical tipping point for the availability of surface area, this transition to overflow representing a limitation on surface area. And then one other thing I wanted to point out. So on both the MG1655 and on this plot, you'll see the experimental data is hovering around 1 to 1.2. You can see our predictions increase as the growth rate gets smaller. So at smaller growth rates, the predictions say there is more surface area available, but the cells don't use it for the electron transport chain. So the question is, as we grow slower, why aren't the cells shifting to a more efficient metabolism? And that is correlated to an interpretation of what we're looking at. So the experimental data is chemostat data. It's energy limited data. And at that inflection point, the cell has the option of expressing higher efficiency electron transport chain components to use that surface area. Or what the cell can do is express alternative substrate transporters. And what we see is E. coli actually shifts to alternative substrate transporters. So it's energy limited. It's doing a bet hedging strategy here where it shifts to alternative transporters with the hope that it's going to acquire another energy source. Then one last analysis here. So these analyses do require assumptions on things like the kcat numbers of these enzymes. The question then is, how sensitive are these simulations to the error that is inherent to these reported kcat values? So with this analysis, what we've done is we've randomly perturbed all of the kcat numbers in the simulations between 5 and 95% of the assumed value. For each of these perturbations, we analyze the situation 100 times. So randomly perturb those values between plus or minus 5% to plus or minus up to plus or minus 95%. Run each of those 100 times, and then we looked at the capacity of the system to generate maintenance energy. And randomly perturbing these systems doesn't have a large effect on the system. The system seems to be robust to these perturbations in that the capacity of the model to generate maintenance energy is quite stable even with these large perturbations. So to conclude, microorganisms interact with their environment via the surface area, and there is a finite surface area based on the physical dimensions of a cell. Not only is the surface area finite, but the membrane has a finite capacity to host proteins. You can't go up to 100% protein. Your membrane would fall apart. So we integrated in both of these constraints into our analysis. With this analysis, our interpretation of mu max is that it represents a Pareto trade-off between the capacity of the system to generate macromolecules and maintenance energy. So the cell runs out of surface area, and at that Pareto front, that defines its mu max value. The membrane surface area that's available for central metabolism enzymes, so these are just the central metabolism enzymes, seems to be somewhere around 7%. Overflow metabolism can be predicted with this analysis, and this overflow metabolism represents a point in the phenotype where the membrane surface area becomes critically limiting. The cells shift more of their resources to glucose transporters, less to the electron transport chain. And this analysis also suggests that the submaximal PO numbers that are often seen in E. coli cultures are an optimal strain or an optimal strategy where the cell is maximizing a finite resource, in this case, membrane surface area, at the expense of energy conservation. So quick acknowledgements. So Ashley and Tomas are Campbell and Adrian, our colleagues here at Montana State. Will Harcombe is a colleague at Minnesota. Mauricio and Krishna are colleagues at Toronto. I also want to reinforce Jürgen's message. So again, metabolic pathway analysis coming up this summer in Vienna. It would be great to see you guys there. And with that, I would be happy to take any questions.